It's good to see you after such a long time. I think.
Yeah, it is. Good to see you too. I was telling Suchi.
Good morning to everyone on the second day of International E-Workshop. Hope you all are ready for the roller coaster ride for understanding, grasping, and improving your skills. May I request Dr. Yasha Hasija, Associate Professor and Coordinator of the workshop, to please introduce our first experts of the day, Dr. Samik Ghosh and Dr. Sucheendra Kumar. Ma'am, please.
A very good morning, everyone, and welcome to the second day of the International E-Workshop on Bioinformatics. I am pleased to introduce you all to the experts of the session, Dr. Samik Ghosh and Dr. Sucheendra Kumar Palaniappan from the Systems Biology Institute, Tokyo. Dr. Samik Ghosh is a senior researcher at the Systems Biology Institute, Tokyo, and serves as the CTO of SBX Corporation, leading the software development effort of computational platform, device consultancy, and drug discovery pipeline consultancy. He received his bachelor's in technology from India in 2001, MS and PhD in computer sciences from the University of Texas at Arlington in 2004 and 7 respectively, where his research was focused on computational modeling of biological systems. Dr. Sucheendra Kumar Palaniappan is a seasoned technologist and scientist with years of practical problem solving and software product development expertise for tackling challenging problems at the crossroads of computational and complex systems. He's worked on both applied and basic research in employing large-scale data analytics, machine learning, text mining, and dynamic modeling techniques for deeper understanding of complex systems in biomedicine, socioeconomics, and related areas. Currently, Dr. Palaniappan leads the data sciences and engineering teams at SBX Corporation as its Director of Engineering. He also serves as the CTO of SBX Technologies Corporation and adjunct senior researcher at the Systems Biology Institute, Tokyo. He received his Bachelor's of Engineering from India in 2006 and PhD in Computer Science from the National University of Singapore in 2013. I invite both the speakers to take over their session on creating an engine of scientific discovery, analyzing text in biological context.
So thank you, Yasha, for the kind introduction, and good morning to everyone from Tokyo, Japan. It's a pleasure to connect with you all. My name is Samik, and I will be leading the first part of today's session on creating an engine of scientific discovery. I have shared my screen. I hope you all can see it.
Yes, sir, it's visible.
Thank you. So the way we want to do this session today is I will start the session by giving you all an overview of what is it that we mean when we say we want to create an engine of scientific discovery, and why is it important in the context where we stand today. And as you can see, the focus of today's session is essentially on analyzing text in a biological context. And to drive that part of the session on creating an engine of scientific discovery, where we focus the lens on text, I would ask my colleague Suchi, as he's known by, to drive that part of the talk, where we will also have some hands-on session for you all to get started. So for the first 40 minutes or so, I will be sharing my screen and then hand over control to Suchi to drive the rest of the discussion, where we will be focusing on understanding text in biological context. So I'll skip my introductions, which Yasha kindly provided. But to just give you all an overview of why we want to talk about this today, we have spent decades of research at the intersection of computing and biology under the umbrella term of systems biology, with a focus on healthcare and drug discovery. And what we have realized with over the last almost 20 years of research is there are fundamental limitations in the nature of human cognition, in being able to understand and make sense of the complexity of life as we know it. And artificial intelligence, or umbrella term of AI, can potentially be a solution to be able to augment the intelligence of humans and overcome some of our weaknesses, and therefore allow us to be more efficient and organized in our scientific discoveries and contribution to society at large. So, as part of this, creating an engine of scientific discovery, this cannot, of course, be done by a single individual or a single group or an institute, be it in Japan or in India or anywhere in the world. So, what is needed, we believe, is to be able to actually have a framework or a platform which will enable researchers across the globe to be able to contribute towards creating an engine of scientific discovery. So, what we will do is we will give you our small effort in creating a base platform called Garuda, which is a connectivity and automation platform. And then we will delve deep together into studying one particular area which for all of us as students of science is of huge relevance, which is understanding text, since most of our scientific communication is using text, be it in publications, in research papers, or in blogs. And I think this month we are celebrating a major milestone, as you probably are all aware, where DeepMind came up with AlphaFold and essentially changed the game in the Caspi competition, and provided a solution to a grand challenge in biology. Now, what are the implications of that in terms of actual contributions to drug discovery, to actually affecting patients in real life scenarios, still needs to unfold. But as scientists, we are excited by the fact that this combination of machine learning, large data sets, and new technologies has enabled us to do a paradigm shift in terms of pushing the envelope and taking the field forward, at least in the area of protein folding structure prediction. So, as I mentioned, the vision of our group is around creating an engine of scientific discovery. It is led by a position paper that our president of the institute, Dr. Hiroaki Kitano, posited around 3 to 4 years ago, where essentially he said that can we actually leverage the power of artificial intelligence together with other technologies as well as the scientific mind to be able to create a new paradigm of scientific discovery. So, as I said, we are from Tokyo, and just to give you all a background of our group, it's a very diverse group of people who are focusing both on fundamental research as well as applications of computational techniques in drug discovery, in healthcare, working with patients in a clinic. And people come from diverse backgrounds of computer sciences, biology, physics, because as you can understand, it requires a village to work together towards creating an engine of scientific discovery. And the theme of the research is around an AI for science, which includes both the development of tools and platforms for leveraging new discoveries which are happening in the computational side and bridging the gap to their applications in life sciences and applied research in healthcare. And as I mentioned, that our small contribution towards this effort is through a series of platform technologies that we have developed. One of them is called Garuda. Uh, it's a connectivity and automation platform to enable next generation of bioinformatics and computational biology tool development, and I'll talk very briefly on that. But the major part of this today's talks will be focused on text analytics, because as I mentioned, text is a very core part of the scientific discipline. And therefore, what can we do to leverage new developments in natural language processing, text mining, to enable scientific research, specifically research in the biological sciences? Now, what I want to do is actually for the larger audience and students who are involved in today's session is to drive you a little bit through the history of the genesis of scientific discovery and why we may be at a very unique juncture in time to be able to change the paradigm of scientific discovery and take it to a next level to enable research specifically on biological sciences. So, of course, when we say scientific discovery, it is not just in the biological sciences, but because we come from a background where of bridging computing with biology, we are focusing on what we can do in the field of biological engineering. So I want to start by giving you, as I said, a little bit of the history. So maybe a lot of you are aware of of course, all of you are aware of the Nobel Prize, but some of you might be aware of an equivalent in computer science called the Turing Award, named after one of the pioneers in the field of computing, Alan Turing. And there is a well-known test in the field of AI in computer sciences, which is called the Turing Test, which essentially asks the question that if we have a session between a sufficiently intelligent computing machine and an interaction with a human, can the human detect that it is not interacting with a human on the other side, but indeed with a sufficiently intelligent computing machine? So that was the Turing test, and what we have posited as a crazy goal is whether the Nobel Committee can give AI system a Nobel Prize without noticing that it was actually discoveries which were driven by an AI system and not just a human scientist. So that's kind of a big goal that we wanted to drive the vision with. Whether we reach there and whether it's actually a combination of AI and humans, we don't know. Maybe all the students in today's session, when they come into research in next 20, 30 years and contribute to the field, will be able to define it better. But if you think, as students of science, if you think of what is the process of scientific discovery, if you read a little bit about the history of scientific discovery, you will see that a lot of it is actually driven by what is called serendipity, right? Which is finding or stumbling across something by accident. And a lot of it, of course, is driven by our unique characteristic as humans of intuition, specifically in this context, scientific intuition. So if you look into the history of scientific discovery, you will see that beyond systematic processes, a lot of actually paradigm shifting discoveries were driven by serendipity. By accident, they stumbled upon something and came across it, or by actually driven by a unique intuition of a scientific mind to be able to actually come up with the problem. Now, what we are seeing is, with the explosion of the data and the technologies that all of you are working with on a day-to-day basis, not just in science but in society in general, a lot of these cognitive problems are actually coming up in scientific discovery. So I've listed a few of them. I'll not go into all of them in details, but I'll pick up a few to help you all understand what we mean by the cognitive problems which have come up in modern scientific discovery, where we are suddenly having a lot of data, a lot of computing resources available to us. So the first problem, which I think all of you will appreciate as students of science, is what we call the information horizon problem. So I think if you do a Google search on number of scientific publications in a year, I think you will come up with roughly around maybe 2 million papers per year across all the disciplines. If you look into even a very specific discipline, let's say cancer, and within cancer, let's say you're focusing on immuno-oncology as a sub-discipline also, you're probably looking at, so 1.5 million to 2 million is roughly 5,000 papers a day. If you look into a very sub-specific field like immuno-oncology, you'll probably end up with 100, 120 papers per day. So it's practically impossible for us as humans with our cognitive limitations to be able to actually, even within a subfield, be able to read all the papers, or forget about reading, be able to read the abstracts and understand which papers are of importance to us, let alone actually digesting these papers and going through a scientific process to be able to actually contribute based on the research which has already been done. So that's actually becoming a major problem because the information horizon is no longer visible to us. And that's a major issue. Related with this is another of these cognitive problems which I picked up, what we call the minority report problem. So essentially, if you think of a certain field and you are reading a lot of papers as part of your review of the field, and you find out that 99% of papers, let's say, are reporting that A activates B, while there's 1% which is actually having a contra indicative report and reporting the opposite, right? That actually A does not activate B, A is inhibiting B. Now the question is, again, if you combine this with the information horizon problem, first of all, finding that 1% is like finding a needle in the haystack, because you will automatically be actually given reports when you search, when you look at publications like Nature, on the 99% of the report, which is the majority of the report which is coming in. So then the question comes that, do we ignore a minority report, or do we actually have to examine the quality, like why it is actually giving a contraindication to what the general field is saying? And it may be actually all reports of that A inhibits B are from one single lab, and there may be problems there. Or if there are diverse reports, uh, and all of them are actually saying A inhibits B, it's just that in the total statistics they are only 1%, and therefore a minority report. It may be important to actually look into it, because as I said, I mean, life is complex, and understanding biological processes is a complex process. So just because 99% of reports indicates A activates B, it actually may not really be what is happening inside a cell or the process under which this was reported. And all of you are aware of what is also known in science as a reproducibility problem, which means a lot of wet lab experiments which are reported even in so-called prestigious journals do not stand the test of reproducibility, which means other labs are not able to reproduce exactly the same results, even if they follow the methods, materials and methods in the paper. So if you think of these two, information horizon and a minority report, these are real problems that we as scientists face every day, of being able to actually keep track of the horizon of our research, and being able to actually find out papers which are basically talking about the same thing and therefore pushing the field, or maybe giving contraindication which we might need to actually delve into details. And add to this the whole nuance of cognitive bias in humans, right? So you all are probably aware of various kinds of cognitive bias which is there in reasoning, anchoring bias, availability bias, which is based on the 99% information which is there. There is also a confirmation bias. Again, it's related, maybe 99% of the papers are saying A indeed uh, activates B, therefore maybe what I'm doing in my lab is wrong. So if you think of all the problems that I said in terms of at least the information horizon or the minority report, if you combine this with an inherent bias which we have, then it can lead to a decrease in the throughput of scientific processes, right? So we essentially are not able to move the field forward at a pace which should be possible with the data and the computing power that we have today. So in that context, we might actually want to redefine scientific discovery. So if you think of scientific discovery, it is fundamentally a search and verification of hypothesis space. When we do our PhDs, we are always taught that to ask the right question, because if you ask the right question, you will be able to come up with a solution. Even if it is suboptimal, you will be able to contribute to that question. But maybe we need to actually also now redefine and rethink in this context that our search space itself is limited because of the information horizon problem and our cognitive biases. And what you see in the blue circle may be the scientific truth, right? That is out there. It's just that our biases or our inherent limitations will only limit us to the red circle, or maybe to an orange circle. So the question is, can we actually enable the expansion of these circles uh, to allow a massive search and verification of hypotheses? So we have to ask the right questions, because we have limitations of time, let's say during our PhD, limitations of our resources available, and therefore getting the right question, getting to a particular point within this circle is important to be able to come up with a meaningful discovery. Now, if we take those barriers out, or try to remove those barriers and enable a massive search of the entire knowledge space, hypothesis space, and have tools to quickly do verification of the scientific process, we may be able to redefine scientific discovery. So then the question that we are asking is that how do we then enable scientific discovery, right? So of course, if you think of autonomy and intelligence on the y-axis and the task coverage on the x-axis, in order to reach towards the goal of a noble Turing, would be on the top right corner of this graph, right? But what we can do today to enable us as scientists is actually help in having connected research laboratories. So laboratories, and we'll talk about this a little bit, about enabling and reducing the barriers in our current workflow so that we can move faster in terms of searching for problems and coming up with verification techniques, which is either doing wet lab or doing verifications by building computing tools to verify the hypothesis. So that's this part where we can contribute, and towards the goal we can also actually have an augmented intelligence, right? So an assistant of AI which is helping us as scientists in the process of scientific discovery. So that's the way we can think to address this problem of creating an engine of scientific discovery. And if you think where we can go in terms of what we will need, what are the tools that we can provide to ourselves as scientists towards this journey, you can think of multiple layers, right? So, for example, a basic functional layer is the ability to connect and automate data and analysis tools, which we are anyway doing today, but they're really disconnected. I'm sure all of you are using some R code or some Python script or some library to do one small part of your analysis, maybe gene enrichment analysis or pathway enrichment analysis, or doing a clustering algorithm and so on. So if you are able to actually connect these and automate these pipelines, that's a good functional foundational module. On top of that, if we can actually have basic AI for task specific AI, or modules, or intelligence or learning modules. So for example, the ability to very quickly read the text and find out connections in the scientific literature. So that's a task specific AI. Then we can actually integrate those different intelligent modules that we built towards finally going from automation to autonomy, which means we actually have an autonomous AI system or an AI scientist which can go through this scientific process without requiring any kind of support, therefore in an autonomous manner, because nobody can develop the entire engine by themselves. So and these are baby steps that we are taking in this journey. But in today's talk, we are basically going to focus on two of these modules. The blue module, which I'll talk about as one of our platforms for connectivity and automation as a functional block. And we will focus on the one part of the green module, which is text. And we will look into how new developments in natural language processing, natural language understanding, and text mining in general can enable us to use these modules in a more intelligent manner as we go through the scientific literature. So let me start by giving you uh, an example of the connectivity and automation platform that our team with our collaborators have built over the last 7 to 8 years, called the Garuda platform. So the first principle, and that is what we actually wrote. In fact, The paper on the right, which talks about the role of collaboration on top of a platform, was deeply inspired by our collaboration almost 10 years ago with the open source drug discovery OSDD project of CSIR India in 2010. And we had a lot of fun. We did a lot of work together, and we learned a lot also on the power of open collaboration, which should be leveraged on an open platform. So if you want to provide a functional blue module to enable connectivity and automation, we cannot actually have it as a closed platform. So we wrote a paper in Nature almost 9 years ago now, where we essentially said that instead of having small Python, or in those days it was mainly R tools, to allow uh, bioinformatics research, we need integrated platforms which allow these different modules to interconnect and be used in an automated computational pipeline manner. So therefore, we propose that what we need is a platform focusing again on the biomedical field, which needs to be open, which means it should be able to interface with different kinds of tools. It should allow discoverability, which means for young scientists coming in, I'm sure that all of you have written some piece of code for your initial course in bioinformatics, which I'm pretty sure was already there, but you were not able to discover it, and therefore you had to actually build the base blocks again. But if you were able to very quickly discover a base block, you could have spent more time on actually enhancing that base block. So there's a need to be able to have a discoverable platform, and the navigability, which means the interconnectivity, being able to connect, because most of the time our computational tools, we don't use them in silos. We don't use them only one tool at a time. Typically, in any omics analysis or any kinds of bioinformatics pipeline, you would be actually having a lot of downstream and upstream processes around the small piece of code or analytic component or algorithm that you have developed. So the ability to actually have an open platform which allows discoverability and navigability was an important, and we proposed Garuda. So I mean, the left hand side of the slide is not maybe not important to the audience today. It comes from Hindu mythology. It was a bird which served as the Vahana, which is the Sanskrit word for a platform, if you will, for God Vishnu. And what we proposed is Garuda is a connectivity platform. It is not a data analysis platform or a data warehouse or a sequence analysis platform. It has a much more simpler goal, which is to allow different components to interoperate and connect together and allow people to build workflows and automate workflows in a cross-platform manner. And you can see the last bullet. We focused a lot on automation and traceability. I talked in the initial part of my presentation on the need of reproducible science. And therefore, in computational field, if you are able to not only automate our pipelines of analysis, but we are also able to trace what we are doing, right? Exactly what steps did the algorithm go through? How did we do partitioning of the data and so on? It will lead to a traceability of the pipeline, and therefore empower the reproducibility of a pipeline in a different context. So therefore, the Garuda platform is an open platform. It supports different operating systems, because we could not say it's only for Mac or only for Linux or only for Windows. If you really want to enable, if you remember the blue block, a cross-functional module, it should be very low footprint, which means we cannot say that this requires GPUs or very high performance computing servers on which they can be installed. Then they cannot be distributed and used across the entire scientific community. And also on a technology level, what we did was we enabled people to what we call Garudify, or get their existing algorithms and code onto the Garuda platform by providing open application programming interfaces in different programming languages, so that people don't have to again rewrite their existing R code or Python code. they can simply put a wrapper and Garudify and bring it to the platform, and therefore leverage the discoverability, the connectivity, and the automation that this platform provides. So there are four fundamental components to the Garuda platform, which you see here, and I'll show you a brief run through one example and a small homework at the end, if you will. So the Garuda platform essentially has a Garuda dashboard. So you can think of it like your smartphones. You have all the apps which are there. These are all apps which are related to bioinformatics pipelines or genome analysis pipelines and so on. And you also have like a app store which is called the Garuda Gateway on your right, from where you can essentially download what in Garuda are called gadgets. So any software module database which becomes part of the Garuda platform, they Garudify it, essentially becomes a Garuda gadget. And there is, as I said, a whole set of tools which are available to help you to convert your existing software into a Garuda gadget, Garudify it, and make it available on the platform. So all these, as I said, are available freely. And the left hand side is, you can think of it as the connectivity map. So, and this is the key part of Garuda, because if I just give you like a repository of all databases or all tools, then it's just a list you can download and you can use them. What is more useful is, can I actually help you to connect these tools together like Lego blocks and build your pipeline? And in order to do that, there is an intelligence which is behind the hood of the platform, and that's called the Garuda connectivity graph, which actually empowers the building of what is called Garuda recipes, right? So I would not only have different Garuda gadgets which are there, I will also be able to actually connect them together by leveraging the Garuda connectivity graph, which was there in my previous slide. And I can build these kind of recipes or bioinformatics pipelines, which may include some component written in Python, some component written in R, some may be a Windows only component. But since all of them were Garudified and they're part of the Garuda platform, they should be able to interconnect, interoperate, talk with each other, and build a pipeline on that one. So these are typically what you see here are called Garuda recipes. Each of these rectangles that you see on as icons are different gadgets which are there, and they have been connected in a specific workflow by the user. So you can see here, you can build very complex recipes. You specify what kind of data you're giving as input, what kind of data is coming as output. You connect these and you can build them. Now, of course, a lot of you must, if you work in the field of sequence analysis, there is a lot of existing tools which support similar functionality in the context of sequence analysis. What Garuda wants to do is take the same concept of pipelines and automation, and they have been there. I mean, if you have heard of an open source called Nime, which is essentially a pipeline building tool, which has been there. So the community has made a lot of effort in order to actually build pipelines. Now in Python, we also have Jupyter notebooks, which can schedule different pipelines and so on. But what Garuda wanted to do was take the same concept, but actually apply it at one level higher, which means one gadget or a one rectangle here may be a Jupyter notebook of Python, while the other one may be an R Studio package that you are using, because that's the reality of analysis. And then Garuda allows them to be interconnected, and therefore we can build and execute recipes. And since everything is on top of the Garuda platform, we can trace the different activities and we can essentially be able to reproduce them. So you can think, I mean, one of our goals is, just like you have protocols which are submitted for wet lab biology, you can think that why can't we actually submit our protocols or bioinformatics pipeline? So this is the pipeline that we built for this analysis in this paper, and you can download this as a package and execute it on top of the Garuda platform. So I want to end my part before we go into uh, the core functionality of AI module, or the green block which I had shown in my diagram. So I want to end the functional part of it, the basic connectivity and automation, because without connectivity and automation, you cannot have intelligence. So I'll give you one example, and I promised a little bit of homework. So what we have done, if some of you have worked on mass spectrometry, data analysis, Shimazu is a major equipment manufacturer out of Japan who build these mass spec equipments which are used in metabolomics, in proteomics, and in different areas of biological data analysis. So typically what you will do is, this is an equipment you will have in the lab. It is connected with a computer. It has some basic software which allows you to process the raw data. And what you will have to do is, then you can see this tower here. You will actually have to physically transfer this data, because most of the time they are not connected on the cloud or on the internet, so you can't access this PC from other PCs in the lab. So you would actually use a USB drive to transfer that data to a different computer, on which you will be able to actually do a pathway visualization on cake for metabolic pathways, or do a volcano plot analysis of the raw data, and so on. And if you actually are doing a multi-omics or a transomics analysis, which means you're having enzyme data, you're having metabolites, you're having other kinds of expression data which you want to correlate, then this process can be very tedious and takes a lot of time, right? So what we did with Shimazu was, we actually uh, worked with them to build a set of Garuda tools or gadgets which are able to take data from the Shimazu equipment directly in terms of their data files. So it can be metabolome data, proteome data, fluxome data. These are very standard data which are used on on the Shimazu mass spec equipments. And we built a series of gadgets which allow people to do standard bioinformatics pipelines, which they are doing every day, but then they use multiple different softwares, multiple different physically different PCs to do this data mapping the metabolites to the keg IDs, visualizing the experimental data, and so on. So very simple processes, but which actually take a lot of time. And therefore we built, if you remember, different recipes on the Garuda platform. So if you have metabolomics data, you use this path of the gadgets. Each of these rectangles are different gadgets which are there. And then depending on what you want to do, you can simply run this Garuda recipe and be able to generate the results without actually having to launch different pieces of these softwares, worry about transferring the data, and so on. So I'll just show you a video of this process. So what you see here is the Garuda dashboard. As I said, you can think of it like your smartphone. And each of these rectangles are different gadgets. On the left hand side, you can see different categories of gadgets. So I'm going to the Shimazu pipeline. There are 4 gadgets you can see. It launched the first gadget. This is a Windows program which is actually directly connecting with the Shimazu machine. It's loading their sample data. And then it clicks discover, and therefore Garuda tells him that in this recipe you can send this data to a gadget called the data mapper, because you need to map these raw data, the metabolites, the keg IDs and different IDs, onto your pathway. This is a standard process that we have to do. Now this data mapper is a different software. It's written in Java, but because they are Garudified, they're able to talk with each other. And this piece of code in Java essentially took the raw data, took a keg pathway, and was able to actually map the data. Again, you discover, you send it to another software package called Wanted. Now, this is a program which allows visualization of a map data in a GML format, a graph markup language format. So what you've seen here, what I'm doing manually is I'm actually going through this recipe of starting with the raw data processing. I started with the metabolites. You can see NADH, NADPH being actually visualized. Now I'm going through the same process and actually uh, sending a different form of data. The pipeline is same. I send the data. I did the data mapping using the data mapper gadget. And finally, I sent it to a visualization gadget called Wanted. And now you see it also actually has plotted the enzymes from the proteomics data which was there. So this entire process, I was actually able to run it as a recipe on the Garuda platform. Now, if I really want to create an engine of scientific discovery, I cannot be actually going through this 10 or 15 mouse clicks for a large scale study that I'm doing. So therefore, what is needed is I have defined this pipeline, I have checked that this pipeline works okay, and I want the system to be able to actually automate this pipeline. So now in the next video, what I'll show you is the same pipeline, but now it has been actually made into a recipe. It is basically loading the files at the start of the recipe, and it is all the different data which is required to execute this recipe. And then when it says start recipe, it's actually automatically launching, doing the analysis, sending the data to a different software or a gadget on Garuda. And you can trace how this recipe is being executed during the process of analyzing this data set. So the entire process is now being done manually. For this demo, I stopped and actually showed you some of the process which is being done. Sorry, this entire process is now done automatically, right? Which means I don't want right from generating plots to actually getting the results. I am no longer need to do it manually. I can actually open my recipe and execute my recipe. I can even execute my recipe remotely. So I may not actually be having access to let's say a BSL level 1 or level 2 lab. The data is coming in that lab. I directly want, I have set it up so that the data coming from that equipment is automatically processed through this computational recipe. And then once the recipe execution is completed, I can see the detailed view, I can see the recipe logs, and I can download the data and use it. So this is the one example I wanted to show you all what we mean when we talk of the blue layer of providing a basic platform which will allow us to define our computational workflows. And of course, you can think of it not just for computation. uh, for the sake of today's talk, I just focused on the computational pipeline, but you can think of the same thing being done for robotic experimental labs. So the experimental processes uh, of actually doing experiments can be automated through robots in the future. And therefore, these gadgets are no longer software components, but actually uh, robots which are connected and executing this pipeline. So as I promised, this entire pipeline which I just showed is actually available freely. And I have given the link, and I'll share with the team this presentation document for you all to test out. So if you visit this link, you can exactly download for free all the components that I showed. And I would encourage you all, and it comes pre-packaged with all sample files. So even if you don't work on metabolomics data or mass spectrometry data, it's fine. You can just download with all the data. And I would encourage you all to actually try it out and see what are the problems that you think are there in the current platform, which will need to be addressed by the community as we go towards creating an engine of scientific discovery. So, of course, the connectivity and automation layer that I showed is an important and necessary component. But what is really important is to be able to build the intelligence on top of this by leveraging machine learning to essentially provide insights from the data. So I showed you the basic platform which will help us in moving very fast with our data collection, with our data processing. But what is needed on top of that is actually a learning platform. And as I said, we have, again, coming from our Hindu and our Hindu mythology. This framework is called the Gandhara Framework. As you probably remember, Gandhara was a civilization, and therefore what we believe is learning cannot be a software which you can plug and play. Learning needs to be a framework which allows different kinds of learning techniques and algorithms. So this is not like, let's say, if you want to do a deep learning, you can use a platform like PyTorch or TensorFlow. It is rather a framework which supports different classes of learning algorithms. And if you remember your history and mythology a little bit, the capital city of Gandhara was Taxila or Takshila, which was the center of learning. So therefore, our text mining component is actually called the Taxila platform, which Suchi will be talking about. And I want to leave you all with this slide. As I was talking with Dr. Yasha before we started, this year it's almost also coincidentally 10 years when we had the pleasure of actually working with Dr. Yasha and a lot of other people like Vinod and Anshu, who are there in this workshop on what was called the Connect to Decode project. So you see here on right a picture which we had taken of actually working with 200 students to construct a metabolomics map of Mycobacterium tuberculosis. Now, at that time, 10 years back, we needed to actually get all these students read a lot of papers, a lot of protocols were defined to extract data from the full text or from the abstract. And then we were able to generate a hairball map using some of our softwares. One of them was called Cell Designer, which is on the Garuda platform. At that time, Garuda as a platform was not there. And in fact, we were just initiating the ideas of Garuda and writing that paper around 2010. So the question is, this is what we did 10 years ago. A lot of effort and enthusiasm and protocols by the entire team of 200 plus students and faculty who all came together physically. We traveled from Tokyo to New Delhi, spent like 3-4 days at the workshop. Now the question is, after 10 years, with all the developments in AI, in machine learning, in computing, in big data, what have you, what can we do to actually enable such processes to be done at a large scale, leveraging machine learning, in this case, leveraging text mining. So I want to stop here and hand over back to you, Suchi, to explain to the audience that what can we do with text mining today, which we could not do 10 years back when we had started the OSDD platform. So I'll stop my screen share and hand over, Suchi, to you.
Yeah.
Sir,
Uh,
hi sir. Sir,
Hi.
can some participants have some queries? Can we take 1 to 2 queries regarding
Sure.
the presentation? So that
So I
was a
have to open the. So you will drive the queries for me.
no,
You will
sir.
tell me.
I will tell. I'll ask you the queries.
Ah,
Yeah.
okay. Thank you. Yeah, sure. Go ahead.
So it was indeed a great platform for automating different softwares and of different languages. So, sir, some participants are asking that how we can contribute, like if you want to contribute any recipe or any ingredient of the recipe, how
Correct.
we can contribute. Is that to be published or only codes will work?
No, so that's a very good question, and thank you for this question as well. I'm very happy to hear this question. So they can absolutely contribute, and as I said, I'll also share the site and the entire presentation at the end. There is no need for publication, nothing. They have a piece of R code, Python code. We will connect them with our developers. In fact, Suchi's, another part of Suchi's team does this. They will be given freely all the packages to Garudify if they have an R code or a Python code, and they can basically submit their code on the Garuda platform. In fact, I did not specify this, but what is the licensing model, right? What is it when you upload something, like if you upload something to an Android store, there is a certain license that you have to actually have with Google, right? For that matter. Now, the licensing of Garuda is what we call BYOL, bring your own license. So which means you decide how you want if you want to give it as an open source with a Creative Commons, if you want to give it as a MIT license, we will simply take that license and say, hey, on the Garuda gateway, that's the license that you will. So absolutely, everything is available on Garuda Alliance website, and our team will be very happy to get more and more gadgets from the participants.
Thank you, sir. So another query is bit curious, I think. Sir, one of the participant has asked that can Garuda platform be used for industrial scale biomanufacturing or in today's context, industrial 4.0?
So very interesting question, and I'm very happy to hear this. So the answer is yes. For the sake of time, and today I did not want to actually show those use cases, so I skipped those use cases. But what I'll do is, because the question came in, I will share with you all a entire website of videos, and you will see a lot of videos of Garuda, just like what I showed you all right now, where it has been applied in an IoT context, in Industry 4.0 context, and also in the context of biomanufacturing. So absolutely, the participant is right in thinking that the platform can be used beyond the traditional bioinformatics pipeline.
Thank you, sir. These are all the queries. So we can continue.
Great. So
Dr.
Suchi, I'll stop my screen and you can take over.
Yes.
I'll also stop my
Please,
video just to share, save some bandwidth.
please let me know once you can see my screen. Uh,
So I can see your screen.
Yes, sir, your screen is visible.
okay, great. So as Samik was during his talk mentioning that we as a research organization are in the realm of creating an engine of scientific discovery, and today's this part of the talk, I would like to focus more on looking at text or natural language, or the language in which people communicate, right? I mean, and how text can play an important role in aiding biomedicine, and more generally in helping towards creating that engine of scientific discovery. So the way I will organize today's talk is essentially around four kind of segments. The first segment uh, is to kind of give a very broad overview and a quick overview of why text is important to look at, and especially for text to be an important part of how it can aid our biomedical research. The second part is essentially going from this very 100 feet view uh, to motivating some of the problems that we at SBI are working on. These are again problems where the solutions are still not fully out there yet. But then we hope that the audience and the students would be motivated to kind of look at text and look at what is the value that analyzing text and the text intelligence can bring about, right? And the third aspect is how we are taking the learnings and our experiences and research in the domain of text and packaging it as tools and solutions. And we will actually, together with the participants, do a quick exploration of one of our tools called the Taxila tool, as Samik was already mentioning, which we specifically made for the COVID era, where we were trying to see, can we quickly collect all of the scientific communication that is happening around COVID, and then give a platform which will enable scientists to quickly understand what is happening in terms of publications in the realm of COVID. And after this, I'd like to give a very quick primer, where we have prepared a very small programming kind of co-hackathon, where I will basically walk you through that analyzing text is not as difficult as we might think. And I mean, given today a lot of the machine learning or AI is rather commoditized in the sense that you don't have to build that model. The whole idea is how do you solution for your use case a particular library and how you can use that. So I'll be splitting this into four parts, and the first part is kept to be very abstract because the kind of summary that we want to give a 100-feet view is almost 20 years of research that has been happening in trying to understand text. Of course, if participants have any questions, we can of course take it later and as we discuss. So the first part is going to be a little abstract to try to motivate text and its utility. And then we will basically go ahead with that, right? So coming to the first part of why text, right? I mean, we are living in an age where big data and rapid discoveries are the forte of biology, and this is thanks to both experimental advances where you can really run high throughput experiments and producing tons and tons of data, and then the AI or the machine learning or the analysis or the bioinformatics pipelines for analyzing this data have already also advanced, uh, and this is typically what is called primary data, and what a lot of students in bioinformatics primarily focus on, and this has been the forte for a long time. But if you look at text, and I mean, I just want to bring a small kind of an simile, which is if you look at the big tech players today, the Amazon Alexas, or the Google search engine, if you really think about it, the kind of advances that we are seeing today and the monopoly that these companies are able to create is because they have had a very good hang of text. Google started as a search engine where it was trying to index all the web pages. And even today, when you're talking to Amazon Alexa, what it is doing is trying to understand what you're speaking. It's understanding natural language and then trying to help you towards the goal of an intelligent society, right? In that sense, text has been the forte of generally a lot of advancements that you see today in the technology space. And similarly, I mean, if you look at text in the space of biology and healthcare, it is still the lingua franca of science. I mean, all of your publications that you see is natural language. I mean, people do experiments, select the experiments, do the inferences, and they put all of that knowledge into text. And similarly, you have reports, regulatory reports, you have clinical trial data, clinical trial documentation, and also nowadays with a lot of the neurological conditions, people also talk a lot about their their uh, their their conditions in social media. So social media has also become a primary source of data or textual data, which can really help drive science and biology. And this is what I would basically call a secondary data, in the sense that it's not data as we are used to in terms of data that is coming from, let's say, the mass spec equipments that Samik was talking about, but this is data that is consolidated in natural language. Now, the problem with text is it's 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 very noisy, it's difficult to understand, and the the complexity increases uh, in text in the biomedical space. So it's generally a hard problem to get a good hang of text and making strides there. But today, the goal of this whole session is I want to basically motivate and hopefully inspire some of the students out there to basically look at text as a very important and powerful tool which can actually first help driving the process of science towards efficiency as it stands today. But also, if we can somehow get a good hang of text and represent the whole knowledge as we know today, drive new hypotheses generation and new experimental paradigms owing to text being a major aid. And today, my goal is, hopefully, I will be able to impress upon you why text is an important aspect of generally any biomedical research and even in bioinformatics. And so I'd like to basically kind of go back to the slide that Samik was showing, where one of the goals that we have is can AI or machine learning algorithms eventually win the Nobel Prize? And the whole idea here is a very critical part of that is for these algorithms or machines to be able to conduct experiments. Now, before conducting experiments, they have to form hypotheses. Okay, they must say that, hey, look, maybe this is a new mechanism that is not yet known today. But if I do an experiment setup which can explore this new mechanism that I'm hypothesizing, then it could lead to a discovery. Now, to make that hypothesis, a very important part is how does a scientist today come up with that hypothesis is basically they read a lot of papers and come up with an intuition to say, hey, this could be a possible mechanism. And all of that information is today present in papers or databases. And a crucial part of going towards an autonomous agent or an intelligent agent trying to do experiments on its own is to get a good hang of trying to convert publications or papers into knowledge and using that knowledge to drive new hypotheses. And so this is why natural language or the dimension of text is an important and an integral part as we go towards creating an engine of scientific discovery. Now, the whole domain of text mining or natural language processing. So it's called natural language because, I mean, if you look at a computer program, it's basically looking at zeros and ones, right? I mean, and this is like natural as we talk, as we have discourse, right? So this is called the natural language processing. And within natural language processing, there are multiple components. One is, can you first look at the data or text data and do a processing to clean up the data to understand different elements of it? The next level of natural language processing is what we call as natural language understanding, which means beyond just symbols that are there in language, can you understand the meaning that is coming out of it? And the next level of this whole advancement is what people call as natural language generation, which means has a system understood so well of how natural language evolves that it can now also generate natural language, right? And as I said, this field, I mean, this is basically the Googles and the Amazons of today, right? I mean, a lot of the supremacy they have is because they got their act right with respect to how they look at natural language. And as I said, I mean, the field is so vast, it encompasses information retrieval, information extraction, document extraction, translation, summarization, everything that you see today around you in the intelligent devices, in the intelligent experiences that you have once you go on the internet, is all because of advancements in natural language processing. And the diagram to your right basically kind of summarizes this whole thing, where you basically have text coming in, and you also have a lot of domain knowledge. And and this is where it's different for us as people coming from the biomedical sciences, because a lot of how the NLP shapes for a particular domain is also a lot due to the domain knowledge and even the ontologies that we have built over time to help in understanding of the text. And a lot of new algorithms, developments, machine learning, AI are being built for natural language processing. And the whole idea is we basically go from unstructured text to structured data. And by structured data, this is data that can be queried. It is data where, let's say, you are looking at all your sequence databases, and you can add annotations to it, and so on and so forth. And there are multiple applications of NLP on top of it. So this is essentially the field of text mining and natural language processing. And I think over 40 years of research is where we are. And in fact, thanks to the deep learning revolution, I think we have made significant strides in NLP and generally getting a good hang of natural language in the last 5 to 6 years. And the whole idea is now, hey, there has been so much of work that's happening in natural language processing. How do I then take those kind of ideas and principles and bring it to the biomedical domain? And of course, I will also discuss the biomedical domain is actually harder in that sense to kind of conquer than natural language in a more generic setting. So the next slide is basically why or how biomedical NLP, right? And this is actually a snippet that is basically there from a 2001 paper, where the introduction of the paper is what I have copy pasted here, and it basically says what is it that a typical scientist does when he goes about positing a hypothesis and sets up the experiments for a new discovery. Um, so one of the classic cases is, let's say you want to implicate new proteins or genes to an already known pathway or an already known disease, right? And then the idea would be uh, to first look at maybe something related in published papers, look at conference talks, and then saying, hey, or look at a slightly different domain in which this particular protein was implicated, or a different cell line, or whatever that may be, and saying, hey, maybe this protein or gene can be implicated in my new process or a new signaling pathway. And then we would conduct experiments accordingly, right? So if you think about it, reading papers and getting a good hang of what is being said in the papers is crucial to scientific discovery. And today, one of the key things that has changed over the last many years is that you are able to process massive amounts of data and different kinds of data. So nowadays, even in biomedical research, there is merit to ask a question of, hey, can you use health records? Can you use social media? Can you use policy or regulatory documents also into your biomedical kind of research paradigm? And today, the technology is there, the infrastructure is there to do it. And so the question is, by bringing these things together, can we really push the envelope in terms of how research can progress, right? And this is just to give you a little motivation of NLP and the kind of scale in the biomedical domain. So this is yesterday when I was looking at the statistics. So today, just PubMed, which is basically a place which is looking at biomedicine and biology and trying to keep all the citations. 2020, there has been about 30 million citations that are there, right? And Generally, there is an exponential growth that you see year on year. The number of publications keep growing, and as Samik also mentioned earlier in the talk, even if you're in a very small field that you're focusing on, um, it's almost impossible today to be ahead of the curve and say, hey, because and get a good grasp of what is happening. And today, increasingly, research has become interdisciplinary, which means it's no longer that what is happening in let's say immuno-oncology, you only have to read papers in that particular realm. You always have to expand, okay, what is happening in other technologies, and can that basically cross pollinate and help in our research. So increasingly the number of papers or the subset of papers that researchers have to read uh, is increasing, and it's hard to keep pace with, right? And this graph here is actually showing year on year the growth in the number of papers in PubMed. And one of the applications of NLP, and as I said, NLP is not new in biomedicine. It's been over 20 years that people have been applying NLP. And primarily, it was used to kind of add annotations to the databases which are housing sequence information or biological data. And for instance, there is this one interesting a graph which actually shows that how many proteins today or at that time of publication lack annotations. And if you think about it, annotating a data set or annotating your data is an important part of discovering data. So if I'm a researcher, I want to understand or do data analysis, I'm looking for rich annotations so that I can quickly search and contextualize my data. And it's again impossible to basically keep track by manually annotating every single data set that is being today gathered. And traditionally, NLP has played a huge role in trying to bridge this gap by automatically, let's say every publication comes with a particular data set, then trying to use NLP techniques on the publication and automatically extracting annotations. So this graph basically shows that a large part of what was happening up until then was not leveraging on basically the natural language processing. And hopefully with the idea of text NLP and text mining, this curve would very quickly reach the point where we were able to quickly annotate, and today it is a reality. So this was a very old kind of a paper, and today if you see, a lot of the databases housing biological data have rich annotations, and at the core of it, there is a lot of text mining and NLP algorithms that are being used. And add to this, the other complexity in biology is a very recent paper. I mean, last a couple of months ago, is while text is the lingua franca of science, all communications of science happens through text. It's getting harder and harder to read. And there was a research that was done recently where even the way people write papers has become so complex, and even for people within the same field, it's getting harder to understand and interpret research publication. And increasingly you see how text mining and NLP or the value of that is coming through. Again, a June 2020 paper where journals are now saying, hey, why don't you start writing papers so that it is easy for text mining and NLP to take that and use this for discovery and interpretability, so that you can quickly make sense of all this text data, right? And I mean, I will basically motivate with a few more examples from our own research of this particular problem, and you will see that typically in biology and in biomedicine, text data is immensely noisy. And as Samik was also mentioning in the previous part, immense ambiguity, people using terminology in very different setups. There is semantic variation in terms of the same thing meaning very different things, and the dependencies between different components in the text is much more complex and embedded in the nature of in biomedical NLP, right? So that is to kind of motivate why and how NLP or in the realm of biomedicine is important. And as I said, I will quickly go through the next two slides, where the whole idea is to basically give you a summary of areas in which NLP has over the last 20 years seen applications. And of course, these applications are refining. These applications are getting more ambitious now, thanks to a lot of the advancements that are happening in deep learning and in the other side of AI and machine learning. But typically, NLP up until now was basically used to augment the biological data cycle and annotations in databases. For information retrieval, when you go to PubMed and you ask a particular query, is the is the underlying algorithm trying to understand what is it that you're asking rather than just doing a keyword search. There is a big body of work, I think over a decade of work, where text mining was used to do PPI extraction. So trying to look at publications and extracting PPIs from them, and looking at text and tagging these text for genes or proteins, understanding gene disease annotation associations, using text for acronym or term extraction, and even for augmenting databases, sequence databases, and so on and so forth, right? So this was primarily the use cases up until now. And today I'll be also showing and motivating with some of our own case studies, which are related to these, but they are trying to push the envelope one level ahead in terms of trying to use the more AI techniques that have come about in the last many years. And typically, if you look at NLP tasks, or if you're given a piece of text, uh, there are multiple things that you want to do. First is you want to get a good hang of the words, or what we call as tokens. So you want to understand, hey, where is a word boundary? What is the part of speech? Whether it's a noun or a verb, whether a particular word is a protein, whether it is something else, or let's say there are two particular words. Let's say, I mean, in the introduction, Samik said that often people call me Suchi, and then my name is Sucheendra. Then how do you also kind of say that probably both are talking about the same person? So this is also, I mean, a core part of understanding words. Then the next level of complexity is understanding sentences, where there is structure of putting words together and conveying a meaning. And then you want to then put together sentences and go towards a paradigm of understanding paragraphs or documents together. And then you want to then ask questions around document similarity, asking interesting questions on let's say, on what it means for two documents to be similar, and the kind of applications of each of these problems. I'll come to them, but they're all very important. And again, the highest level of NLP maturity is, as I said, for machine translation. I mean, today, for a large part of this particular thing, it's been solved where machine translation has leveraged a lot on the AI, and it is basically all the body of work that has happened to understanding words, sentences, and documents. Similarly, summarization, question answering, simplification, all these are tasks in the NLP domain, and also pertinent to bio NLP domain, right? Again, this slide is to basically give an overall view of the field in terms of who are the main users for whom using or leveraging on NLP tools is most useful. What are the kind of data sets that we have today which we can be leveraging? Of course, this was a while ago, and today you can add the whole social media discourse into this mix, and also a lot of the documentations that is in PDFs and so on and so forth. And again, what are the different system types, or what are the different problems that you're looking at in this space is here. And typically, what you want to achieve is kind of construct this knowledge network automatically, where you're able to you're able to connect. And it's almost like the Google knowledge graph, right? I mean, if you have heard, so Google, a lot of the queries where there is a lot of contextual, I mean, people now magically says, hey, Google understands me. Whatever I type, it gives me a very contextual answer of what is it that I'm asking, even though I might not be asking it the right way. Is because it underlying that is this big knowledge graph that Google has constructed. And essentially our goal is also to get a good hang of consolidating all the knowledge in biomedical domain, is to basically construct these knowledge graphs and connect very different fields, where I mean, if you remember, Samik showed this space of hypothesis discoveries, where today what you're seeing is what a particular scientist comes up with based on what he has read, right? I mean, it's still what that circle of all the possibilities of hypotheses is still small, because it is also where a lot of the skill of the researcher to come up with the uh, the right hypothesis is important. The goal is, can we also kind of make this in a way that with machines and with the help of algorithms, you can look at a much bigger space of possible hypothesis. And this is a slide, an image that I took from a more generic applications of NLP, where I mean, this is typically what the Googles of the world are now focusing on, where they look at immense amount of text on the web. They come up with representations of text, or how you want to encode text, and then come up with intelligent algorithms to look at that text and answer specific questions, and each of them becomes an application that we use today, right? And with biomedical NLP, or bio NLP, we are looking at that moment now, of how this whole cycle can also augment and help science and discovery in general, right? So with this very broad kind of an introduction, I want to go into three case studies, and all these three case studies are from our own research, and we've been working on these for the last 4 to 5 years, where we have gone one step ahead in terms of the applications that I talked about in the traditional settings up until now. And we focus on on slightly the problems at the horizon, where we can kind of use some of the techniques that are already coming out today in the text domain, not in biomedicine, for instance, but kind of take that and see how we can move the field forward. And I'll be focusing on three main case studies. And the goal again today is to kind of just motivate the audience to say the kind of interesting problems that can be solved using NLP. So for some of the case studies, I might go in a little detail, but for some of them, I would like to keep it a little abstract, so that we can spend more time on the other aspects as we go into, right. The first problem that I'll be focusing on is the problem of what Samik was also talking about, which is the 10th anniversary of the OSDD CSIR initiative, where a large group of scientists basically came together to read a lot of literature, and making these process maps, or these maps which represent the whole underlying biological processes that are happening in particular disease areas or processes, signaling pathways, and so on and so forth. Now, as I said, this is generally much harder now because, I mean, to keep this pathway map updated, I mean, you can almost think that in probably a week, the map that was constructed with a lot of sweat and effort is almost outdated, because new discoveries, you need to keep up to date with new discoveries and continuously update this map for this map to be useful for any bioinformatics analysis. So the first part of our of what I will discuss today is our experiences around going from text. If you think about what a particular curator is doing or a scientist is doing, they're actually reading this text, understanding it, and going and drawing these process maps. Right. So we looked at the specific problem and we wanted to understand, hey, what is there something interesting there? Is there something where at least assisted intelligence can help, or what is the potential of a complete automation of going from unstructured text or natural language text to such pathway representations? And this is work that we've done over 3 to 4 years that I will try to summarize. And so I'll first begin by basically the motivation, right? I mean, or the generic process of what happens is, typically you have a lot of these PDFs or publications that are there in the public domain, or that the researchers read or curators read. Then they would basically use a tool, typically like Cell Designer, but there are a slew of other tools, and go and basically draw these pathways. Let's say a signaling pathway or all the pathways implicated in a particular disease. And then they would manually go ahead and once these maps are constructed, they would be annotated by linking them with external databases and so on and so forth. And this particular asset, which is the structured knowledge, is very useful for driving a lot of downstream analysis. So for instance, I mean, once you have this particular representation, it is a consolidated knowledge of all molecular interactions, right? And typically people use it for enrichment analysis. I mean, this is something that is very common in bioinformatics analysis, where you get a set of genes and then you want to do an enrichment analysis on one of the big pathway databases. But if you go to the underpinnings of the databases, most of it is hand curated, the high quality ones. Of course, today a lot of text mining techniques are being used, but primarily used for augmentations or annotations rather than actually reconstructing these pathways. And so it becomes a very valuable asset for that. It becomes a very valuable asset for understanding, doing network analysis. So especially for coming up with, especially in the setting of, let's say, cancer modeling. I mean, you're looking at not a single drug, but looking at multiple drug combinations, because the kind of effect that we want to study is circumvented by other pathways. So you want to understand the network as a whole and come up with hubs or key regulators in this network, and then come up with a medicinal paradigm of how you want to probably come up with a treatment regimen, right? Or come up with drugs. And so that is a lot of these representations are used for understanding network properties. It helps us understand what we are missing from a bird's eye view, where new experiments can be designed. So this is one step towards the hypothesis generation that I said. People can use these maps as an aid to say, hey, this particular mechanism is poorly understood, or let's say I take this particular representation and build a dynamical model and do a simulation, and the simulation is not able to replicate what is happening or observed in the experiment. Then you have to refine, maybe add new connections, maybe do that whole process of more focused studies. And this is the genesis of new hypothesis. And as I said, I mean, usually such maps are also useful for looking at new therapeutic or diagnostic biomarker candidates. And as I said, I mean, the effort with the tuberculosis metabolome challenge was one among many such efforts that over the last 20 years the community has put in, ranging from understanding processes or key signaling pathways all the way now. And there is a community around disease pathways where they're trying to map the specific processes for specific diseases. For instance, there is also the ALS pathway. And I mean, in fact, the last 7-8 months, this community has also risen and is trying to map the COVID pathway. What is happening at the viral level? What is the effect on the system? So they're trying to map this whole thing, right, towards gaining a better understanding. Now, as I said, typically, I mean, this is an actual image of how such things used to be happening at least a decade ago, where it was a huge effort of trying to manage and get this whole thing together. And I mean, dealing with all the kind of ambiguities and the kind of minority report that also Samik was saying that, hey, some reports are saying that, hey, this particular mechanism is true, but in a different setup, that mechanism is not true. Then how does that representation change? So all of this is a very important consideration. And one of the things that I said, that natural language is always difficult. I mean, it's difficult because people write very differently. There is no notion of I mean, some things that is very easy for a human to understand is very difficult for a machine to understand. And just one typical example from the same paper where we have posited the potential of, or the possibility of AI to win a Nobel Prize is this particular sentence from a paper which talks about the export of a particular complex, right? And I mean, key questions still remain, right, from nucleus, because it says it's exported from nucleus to where is the regulatory compound in the nucleus. What are the different forms of this particular complex which are exported? So if you see, I mean, if you look at it from a correctness point of view, there are so many unknowns that are there here. And sometimes it's very, I mean, for a expert, when they read it, maybe it rings a bell immediately, but for machines, unless there is explicit information there, we have to deal with all these complexities, right? And of course, this is just a quick kind of a thing of our research where we noticed that a lot of research that was happening, in fact 20 years of research that was happening in this field was largely driven by a series of what we call as gold standards, which was set up in the NLP community. And it was largely the actual community of curators who are actually using or reading papers and trying to reconstruct these pathways, where largely, I mean, there was a gap between these two. So, and that gap was because there were very different formats that, let's say, the NLP or the computer science community looking at the biomedical literature was focusing on, and actually people on the field who are actually curating, they are looking at a different set of standards. And our one of the key questions we asked is what is happening? I mean, there is still a mismatch. There is 2 decades of work, but still a lot of the automation tools are not at the bench of curators. So we wanted to understand what was happening. And so for that, a little bit of a background is that a lot of these pathway maps that you see, there are specific formats. And again, these formats were pioneered by SBI. So for instance, SBML is a very well-known format for representing these processes. And so there are many other formats too for different use cases of how you want to represent these processes. Everyone has their own merits. There is no consensus on what is the best. Of course, SBML is one of the largest such community that is there in the world, because it was largely pioneered by our tool called Cell Designer, which allows for drawing such representations and also do simulation studies on them. And it's still active. The community is very active for using SBML and Cell Designer at the fore for reconstructing these pathways. So I'll probably just skip this slide, which just goes into the details of what this language is. It's an XML file. I mean, for those of you who know, it's just a computer readable file where what a different components are from a biological perspective are mapped onto this particular markup language, and this is used to basically represent processes, and software tools can import these files and show these process diagrams, right? Now, coming to the specific problem that we were trying to understand is, as I said, there was a lot of work, 20 years of work that happened in trying to um, take these publications, use a machine to kind of digest this information and come up with structured knowledge. And it is still a problem that's not fully solved. And there is a huge community in the bio-NLP community which focuses on this problem. Now, what we observed when we started looking at this, and because this was a pain that we had, right? We as curators who are trying to do this day in and day out, uh, uh, we were not comfortable with this gap, and we wanted to understand that, hey, what is it that is preventing going from or using algorithms that we have today to get to a representation where it has gone through, or a curator, what is happening when a curator is doing or constructing these maps. And of course, there are positives and negatives to what a curator does, and all of this came out in essentially our kind of research. And one of the other thing was, as I said, the community was focusing on a specific kind of a format, which was different from the formats that we were looking at. So one of the first things that we did was basically look at this format and convert it to a format that is more that can be imported to readily available tools, right? And this was one of the work that we did 5 years ago, where we actually said that we will come up with a schema of how this particular NLP outputs can now be compared directly um, with on tools where curators are actually or using as a workbench. And of course, even when we tried to do that, which is just I mean, it's not a very hard problem in that sense, where you're trying to map a format which was used by the NLP community thus far, and try to map with what was happening at the community which is actually trying to curate. But there were challenges, and this came out very easily when we were doing this analysis, is uh, the NLP community, the kind of benchmarks that they had, was not looking or was not understanding the context of the biology that was happening in a detail that usually curators would need, or people who would represent these biological entities would need, right? Similarly, I mean, if you think of a process, you're looking at some proteins or genes and some reactions of regulation and so on and so forth, right? So we found that, first of all, the granularity at which biological species were represented was not good enough, or not or not complete, if you will. Similarly, there were events or reactions which could not be very obviously mapped. There was also under specifications of events or reactions, and so there were challenges about how we could take these tools and make it a possible to make them available to a curation workbench. So this was one of the first observations that we published, and then we asked the more pertinent question of, hey, given all of the body of work of the last 2 decades that we have done in trying to understand or extract these pathways, now if I look at what a curator or a biologist or a scientist who looked at the same set of papers and came up with a map, what what is the difference between these two maps? Because that is an important question that you need to understand or get clues to, so that a lot of these tools can go and really help the cause of trying to construct these maps, right? And again, there is no consensus on whether the map that was constructed by the curator is the best, because there are biases there also. I mean, there are cases where you give the same 500 articles to 10 different top-notch curators. There will be differences in the way the map comes out, slight differences, and for us it was also important to understand what those differences were. And again, the state of the art in terms of NLP algorithms, what were they doing, and where is this big mismatch, right? And of course, there were some challenges when we were trying to do this. One is because, as I said, natural language is immensely noisy. So to be able to match different biological players and reactions correctly, and not get into this over-representation problem was challenging. And we came up with ways of how we could efficiently do matching. And also a very important part was, and this is one of the things that came out, is when a curator or when a biologist is thinking of a biological reaction, he's thinking of a whole process. He's thinking of a connected graph, or with very less disconnections around it. And the whole space of NLP, or the research that was happening, was focusing on a piece while, which means they were looking at sentence by sentence, or at best a paragraph by paragraph, and trying to assemble these representations. But when you think of a curator or somebody who is actually making a process map, what he or she has in the background is basically one coherent connected graph, or a graph which has less, fewer or one or two only connected components, essentially. So I don't want to get into the details of the algorithms and what we did, but essentially the inferences that came out, right? One of the important things that came out is what you're seeing here is a graph where we are showing mTOR NLP is by a machine and one is by a human. We call as like what we want to compare with. One of the key things that came out is what a human does is not just extraction, but active selection. So if you look at the number of proteins or biological entities that are there in a human generated map, and it's 100 times, if not more, right? And so there is an active selection that happens by the expertise of a human curator, which completely the NLP space has kind of not looked at. So there are important differences between how human annotates and how machines annotate. Now, you could argue which is better, which is not, but at the end of the day, we need to be aware of the fact that there is this difference. Okay. And also when we are looking at annotations, I mean, human annotators actually what they do is make complex connections while they are reading multiple sentences, or when they're coming together, right? So they combine multiple reactions and form a single reaction to condense information. Now, what is that level of abstraction at which you condense information is again a call that a human curator is able to take better. And of course, NLP algorithms have not essentially looked at that paradigm. And this comes to the other point that I was saying, that connectedness, right? If you look at the number of isolated networks, that when we looked at this very large pathway for mTOR that was constructed over a year, there were only 4 isolated networks. So it was largely a connected graph, right? But if you look at what was produced by an NLP or a machine, I mean, this basically shows the problem of this space as it stood at that time, right? Which was the number of isolated networks was huge. So you were looking at, and if you actually look at the networks themselves, you were looking at best at a graph which had 250 nodes. So that was the most connected that was there. Most of the graphs were just two nodes connected. So there is so much of noise and basically not having the goal or the ground reality of what is it that we are trying to do in this particular space, right? And one of the things that we did in this space is to try to kind of look at the gaps that are there in the current NLP algorithms, and we actually posited for the community. And in fact, over the last many years, many of these problems are slowly being tackled by the community, and we are, I think, going towards a framework where really NLP can play a big role in extracting automatic extraction of pathways, right? So there were challenges around normalization, which means two particular proteins, um, uh, which is basically the same, but they are just, I mean, AKT1 and AKT-1 or AKT space 1. I mean, of course, this is a problem again, which has been in the works in the community for the last couple of decades, but they are still not up to the mark. And one thing that the community had completely not taken into consideration is when you look at these pathway graphs, a lot of the things was around complex formation, and that was something that was also not looked at in detail. The whole idea of composing pathways and understanding the level of abstraction was something that needed to be looked at. And again, the broader question was evaluation with respect to human experts, how good that is, how good that is not. I mean, there is that whole paradigm. And maybe I just missed it, but one of the things that we also show in this diagram is with so what you're seeing here in the background in the white is the human curated map, and on top of it, the green boxes means the NLP system was able to map it, either in terms of understanding that there was a species there or there was a reaction there. And as we increase or we have parameters to make it very strict in terms of how we want to compare, or we lose in terms of how we want to compare. And this diagram actually shows that indeed that when we are very strict about how we do comparisons, it's not up to the mark. But, and this is also owing to a lot of the things that we saw as gaps, is once we then go into the space of trying to loosen it and increase the parameters or loosen the parameters, we are indeed getting a good coverage, right? And again, this is still an open problem, and I mean, more and more good people coming into this space to work on this is very much appreciated. So this was one paradigm when you are looking at text and going to automatic pathways, right? So that's one space of problems. Again, the solution is still out there. There is no there is no I mean, this is an unsolved problem. There has been great progress that has happened in the last 3 to 4 years. In fact, DARPA has strategically come up last couple of years with the big mechanism project, where they were trying to understand and funded a lot of this research in this space, and a lot of researchers around the world are making good progress here. Now, the second part, and again, this is a new application. As I said, I mean, I was trying to focus on applications which are new in terms of the horizon that is there. And this is another study in our research. And this was where we were trying to see, can I directly go from data or unstructured data to insights? In the previous step, you were looking at coming to a point where you have structured data, and the structured data represents a good understanding of the background, right? And for this, the basic motivation was the following. So if you look at the diseases of the brain or the neurological conditions, it's still in its nascency, I would say, to understand the molecular underpinnings of a lot of diseases, and all of them have a huge behavioral or a psychological aspect, even for diagnosis and even for treatment, right? And especially for diseases in this space, while it's great that science is doing a lot of strides in terms of making progress on the molecular underpinnings or mechanistic underpinnings, there is a lot of experiential data that people go and share in social discourse, where people who have certain conditions, they go and say, they join self-help groups, they go to blogs, they talk about their condition, what helped them, what did not help them, and so on and so forth. And the kind of applications in this space is from all the way from subpopulation identification, drug repositioning, target identification, that is coming from experiential data, right? And what we did in this particular study was basically try to combine scientific discourse and the non-scientific social discourse together. And the question was, is there something interesting that comes out in terms of a new insights that can be drawn about different disease conditions? And again, this work is also relevant for rare diseases, diseases which does not have a big population which suffers from the disease. So there is no incentive for pharma to invest in a lot of fundamental research. In that case, can we use power of NLP and text mining to basically say that, hey, if you look at related disease to this particular disease, can I repurpose and look at treatment strategies which were used there, which can be reused here, and so on and so forth, right? So for this, we looked at a class of diseases around depression, autism, schizophrenia, and more importantly, at a rare disease called the Tics disease or the Tourette syndrome, right? And our goal here was basically just look at all of this data to understand whether each of the different data types, so by data type I mean publications as one source of data, blogs as one source of data, let's say Twitter as one source of data, whether it was giving different kind of signals, and whether we could combine all of that signal towards new hypotheses of what is happening, right? And again, this is coming to large scale data that we were able to collect. And what we did was essentially across three domains. One is clinical trials, scientific publications, and actually going and getting data from blogs, discussion forums, and so on and so forth. We were able to collect over 1.5 million articles related to each of these four dimensions. And then we constructed a pipeline where you were looking at this data, applied a lot of NLP algorithms. I will not go into the details of them, but the idea was, can we kind of look at this information, extract concepts or topics that are very thematic for every disease type, and compare and contrast these disease types, right? And of course, we use text mining to kind of do the most rudimentary analysis of frequency analysis, where within a Tourette syndrome data, all the text data in Tourette syndrome, which are the other diseases that are being talked about. Let's say ADHD is talked about very closely with Tourette. All these, by the way, are synonyms of Tourette. Tourette, tic disorder, all of them are same. OCD, for instance, came about. Depression was also implicated together. So the idea is that, hey, some of the diseases that we are focusing on are talked together with Tourette. And again, the question was, which are the genes that have been implicated or talked about in Tourette in the scientific discourse, and is there anything in the blogs or publications? Similarly, drug compounds or chemicals in that space, right? So we tried to do this for each one of the specific disease types that we had, and very interesting insights came about. Of course, I cannot get into the details of them, but I mean, I just want to motivate the kind of analysis that we can do almost in a way that would be impossible for to be done without NLP, right? And again, one thing I wanted to bring about is essentially this picture, which kind of summarizes in a heat map. I mean, folks are more used to heat map in the data crunching domain. But what we did here was we extracted important concepts, which means if you're talking about, let's say, a particular festival like Diwali, then you talk about crackers, you talk about sweets, everything together, and that becomes a topic, right? And every dot here basically talks about a topic. And this is compared across the four disease types, and combining both scientific and non-scientific discourse. Now, of course, what you see here as blue region is basically, I mean, just to say that indeed there is a high similarity between or within the diseases. But what was interesting was the red spots, essentially, which basically brought about the key differences in terms of what was happening in Tourette or schizophrenia or Tourette or depression. And this representation also kind of shows that when you basically do this kind of unsupervised way of trying to look at these diseases, perhaps Tourette and schizophrenia are closer in that sense to each other than the other disease types, right? So this was again something very interesting. And again, we could construct these networks of topics where again clear clusters of the different disease types came about. But what was more interesting was this cluster that was in centered, where this could be the space where we need to look for how things or treatment strategies for one domain can be repurposed for another domain, right? So this is another application of how we used text and text mining to essentially come up with new hypotheses, uh, where the key paradigm that was different was we were trying to get all forms of text, because most of the studies that you do looks at specific corpuses of data. I mean, there is a merit to choosing specific corpuses of data too. But we wanted to see whether just brute force taking all of this data has merits, and we indeed showed that there was characteristic differences between the different disease types, which came out just by NLP analysis, right? Now, the third motivation, and this is motivation that and the work that we have been doing now. I mean, this is work that we published this year in a couple of the premier AI conferences in the world, uh, the idea was again going back to what we were discussing, which is, hey, I have all of this knowledge that, let's say, I find a way to represent. It could be a knowledge graph, it could be some other representations that you could have. Then can you take that representation and basically generate new hypotheses? By new hypotheses, I mean potentially new discoveries that, when validated, could lead to when new experiments being conducted, could lead to new breakthroughs, right? And basically for this particular work, what we did was we encoded this whole knowledge in text into a knowledge network, where every node of this network, if you will, represents concepts or keywords like proteins, genes, and a link between these keywords or concepts means that there is a direct implication where both of them have been studied together, either in terms of different relationships. So for instance, I mean, this could be a graph where, let's say, A and B are diseases, two disease types, and let's say it is already known that X as a drug cures disease A, right? And then from a previous analysis, just like we did in the previous study, I'm able to establish that, look, disease A and disease B are very similar in terms of how or what they kind of their underlying mechanism, let's say. Then a hypothesis would entail for the machine to say that, hey, by inference, can X be repurposed for B, right? And this is what we call as a hypothesis, which can be then tested, right? Now the whole idea here is again to look at data at its full throttle. And as I was saying that if you look at knowledge as a network, these networks evolve every year. So the network that was that looked a certain way last year, the network would no longer look the same. It would have evolved. There is a lot of dynamicity in the way this network is evolving. Some big contributions change the whole properties of the network. And the idea is, or the way we posited the problem is essentially predicting new links given a network. So if you're looking at, let's say, a future network that looks like this, and today's network which looks like this, then how do you combine NLP and also graph algorithms to predict the most possible links, which amounts to a hypothesis? And again, we use a pipeline for NLP for coming up with new hypotheses. Again, I won't get into the details of the whole algorithm. I'm happy to share it after this talk if anybody is interested. But we basically posited it as a temporal graphlet and the evolution of temporal graphlets with supervision, where the next 5 or 10 year data is used to supervise how the network will evolve to the next 10 years. And the hypothesis generation would be given at a particular year. Can I make discoveries, or can the links that I described from my model is indeed the ones where there were discoveries made, right? So this was how we formulated it. We had an RNN and graph learning algorithms to basically encode all of this information. And I want to bring us to basically the experimental results, or what we essentially found. So we looked at PubMed, three domains of PubMed data. So because we were looking at 30 million articles, and in the spirit of trying to understand domains, we kind of split it into three domains. And we posed the question as saying, hey, if I have data from 1950 to 1959, can I make discoveries that happened between 1960 and 1969? Or eventually there was a discovery, right? So that was the way we posited the problem. And when we looked at our approach, which is the last row that you see here, we were better than most of the current approaches. I mean, specifically, they were not solving this problem, but from a graph embedding problem, if we took those algorithms and just used them. So this is also the place where we said, hey, there is a lot of work that has happened in this space. I'll take some of them and just use for our context. But it was not the best algorithms out there. And we came up with a strategy that was much better than that. But what is very interesting, and this is what gives us hope towards building this engine of scientific discovery, is if you look here in this table, you're having 5 rows. Each of them talks about a particular connection that was eventually discovered. So for instance, the relationship between Raynaud's disease and fish oil was made or discovered in the year 1985, right? And what we did is, if we used all of the articles until 1970, where Raynaud's disease or fish oil may have been separately discovered, and asked what is the probability that in the next 10 years these two will be connected, it is a very low probability. But eventually, the case that it gets connected goes into high probability, which means, and the rightmost column here is telling you the actual year when there was a discovery made, right? So for instance, if you look at the last one here, which is schizophrenia and PA2, and the actual discovery of the linkage happened in 1997. And if you look at our model prediction, we could predict that with a high possibility. I mean, I want to remind that we have no knowledge, or our algorithm has not looked at the publications here. We were able to predict that with a 93% probability that these two particular pairs of concepts would eventually get connected or implicated together, which amounted to a new hypothesis, right? So this is basically very, very exciting and interesting, because what this does is, we have made the first baby step towards can we go towards an automated paradigm, or I mean, at this point it is even assisted paradigm of scientists being able to come up with hypotheses that fine tunes their intuition. To basically give more power to their intuition, whether these models can help. Again, we have made baby steps here, and we are actively working on this as a research area. And in fact, we also look for very bright PhD students or postgraduates in computer science, data sciences, bioinformatics, who have a very strong mathematical or a computational background to join us in solving these problems. And again, a couple more slides where we show that So the interesting thing about our approach is also it tells us how we get from one concept to another, because it's a graph. The fact that two concepts get linked and a new discovery can be made, it also shows us how that particular discovery can be made, right? And this shows that two concepts which are three hops away, and so on and so forth. Of course, this is very early work, and a lot of the things that we said here was from an pure graph embedding point of view. Each of these hypotheses have to be very carefully looked and see whether it indeed makes sense, and so on and so forth. But I think we have started making the first headway.
Excuse me, sir.
Yeah.
Can I interrupt you for a second?
Sure.
Sir, participants are saying that the slides are not moving, sir. It's stuck on the hypothesis generation.
So can I try again? I'm going to stop sharing and reshare it.
Yes, sir.
Is it okay now?
Yes, sir.
Sorry, I think I went quite a bit ahead in terms of
Yes,
what I
sir.
wanted to say.
At
But
first, yes, sir. Yes, sir. Continue.
the whole point here that I was trying to drive, and maybe I will just drive it through this particular slide, is what you're seeing here is essentially two concepts or pairs which were eventually discovered. Right? They were discovered that these two particular concepts were linked together in 1997. Okay, and our algorithm or our AI module, we did not give it papers in this space. So we trained on papers until 1970 and said, hey, can you come up with new hypotheses? And it said that the probability of these two concepts, which was, let's say, schizophrenia and PA2 being implicated, was the highest in the 1990 to 1999 kind of a space, and there was a 93% of that happening. So which means incremental knowledge as it is building, paving towards scientific discovery, came out very well in our study. And basically this is, as I said, very interesting and exciting, because what this says is that we are able to at least now have a formal way to understand what it is to make new hypotheses, or what it is for machines to understand making new discoveries. As I said, we've made very baby steps, but we've made the first few steps. And again, in this slide, what I wanted to show was two concepts which were different and got eventually connected, or there was a hypothesis implicating both of them. What was the path to get to the other concept? So this is also interesting because it also shows you how the flow of information or flow of uh, mm, mm, this the hypotheses could have basically happened. And it is here I was saying that a lot of the analysis that we show here has not been biologically validated in that sense. But we look at indeed that these connections have happened in a future graph. And the whole idea now is to work with the scientific community to kind of refine this framework, to kind of go towards a hypothesis-driven framework for science, right? So with this, I'll probably take a little pause, and because we have basically given an overview of text mining and NLP. I have introduced three problems or research problems where really I mean, the field is being pushed ahead, and the research is being pursued, and just to motivate very inspiring young scientists to kind of work on it, and also for very interested students to see if we all can work together. But the next part is where I go into a little practical side of things, where you say, hey, okay, all this is great, NLP is great, text mining is great. Now, how do you then take that and build into solutions or products that is helping me today? to kind of help me quickly understand or assimilate knowledge as it is there today, right? So, and as Samik said in the previous introduction, so we have our text mining solutions, the platform of Taxila, where we do research on text mining, but also try to bring these text mining concepts to the community, to people who can use it to derive novel insights, right? So for this, I'd like to basically give a small walkthrough of a Taxila that we built specifically for COVID. And after this walkthrough and after this talk, I would basically ask students to also register for the COVID Taxila, and also look at what all we can achieve in Taxila and how it can help us delve into the space of text, and how we can draw insights better using text mining. So this will be the first maybe 15-20 minutes of what I'll be talking. And then, as I said, I'll be taking you into a quick primer so that you can just get your hands dirty in terms of just analyzing some text. And for this, I'll be using a Python Jupyter notebook. I'll be sharing the code, ways of how you can install this code on your machines and start playing with it. Because as I said, I mean, in the initial part of the talk, I mean, a lot of the text mining tools are commoditized. You need to only understand what they mean. Using them is just a click away. So if you're motivated by the right problems and how you want to solve these problems, putting them together as a solution is at least now very easy, right? So these are the two components that I'll be focusing on. So for this, I'm going to quickly go to my Chrome browser. Are you still able to see my screen?
Yes, sir, but it's on tools in action wala slide.
Okay. So I need to again stop sharing and reshare. Okay. Are you able to see my screen now? Sorry.
Yes, sir.
Yeah. So as I said, I mean, let me get into the demo, or just a walkthrough of the whole Taxila for COVID. But I want to maybe spend 5 minutes on the whole of what is it that we are trying to achieve with Taxila. And again, our goal is to go from text to insights, or putting your text in context, or taking text and convert it to knowledge, which does not require a lot of manual effort, but with the help of NLP and AI, can we go towards the framework? And as Samik said, I mean, the name of Taxila is from Taxashila, which is the center for learning in the Gandhara civilization. And the idea here is again that our engine actually combines everyday aggregation. So our system every single day goes and picks up all information relevant to a particular topic. In this setup, relevant to COVID, in terms of scientific publications. And you'll be amazed by the amount of scientific publications that have started flowing in relevant to COVID, or relevant to viruses, and generally over the last 6-7 months. And so the point here is, you don't look for data, data comes to you. Our engine automatically goes, looks for the latest clinical trials, looks for the latest publications, looks for the latest archives relevant to COVID, and brings it into the system. Once it brings it into the system, we apply a lot of the NLP or text analysis algorithms that we were referring to in the last many slides, and then use that to for powerful visualization and drawing insights, right? So I'll go over each of them, right? And again, I'll leave this after the talk. I'll basically give access to all our folks who can go into the covid19.taxila.io. This is a website where anybody can go and register. I'll walk through how you need to register and what are the steps to be followed. And then once you register, how you can start looking at text and trying to make sense out of text, right? So let me not spend much time on slides, but basically go into the system itself. Please let me know whether you're able to see the system.
Yes, sir, we are
Yeah.
able to.
Okay. So what you're seeing here is on a Chrome browser. I just opened covid19.tuxila.io. And for those of you after this talk who want to register, you basically go and say you're a new user. You register with your email ID here. You would then basically get an email where you would say that you have been successfully registered to Tuxila, and you will basically get your credentials with passwords. So once you get your credentials and passwords, uh, so in this case, I'm going to log in with a pre-existing credential. Once you log in, what you come in is like a workspace where if you see every single day, all the articles that have been published in the COVID space are automatically extracted. So for instance, until yesterday, every single clinical trial that was published has come into Tuxila. This was published yesterday, right? So, and the way we do that is we kind of organize content into different headers, right? Or channels as we call it. So what you're seeing here is clinical trials, archives. So you will see that because of the speed at which research is progressing, at least in the COVID space, a lot of people are publishing in archives because there is no time to essentially do a peer review and so on and so forth. So in that space, what is happening is a lot of work is published in archives. Similarly, publications are coming in. And a lot of data has been made open in terms of virus research or the general coronavirus family of viruses. The research that has been happening over the last many years has also been made public. So this is a place where as soon as you log in, you are able to see every single article coming into Tuxila. Now, let's say there is a particular article that is interesting to you, then you can just read it just like you would normally. But the interesting thing here is what Tuxila does is it has automatically read this article. It has automatically read this article and found out that, look, it is talking about these different concepts. It's talking about some kind of funguses. It's talking about some pharmacological drugs, right? And then when you click on it, it's going to say, okay, what are the different drugs or the drug terms that are being talked about, right? So it is automatically helping you summarize by identifying key concepts within every article. Now you are reading this article and you are saying that, hey, I want to understand which are the related articles. Again, Tuxila helps you by looking at or computing which are the articles that are related to a particular article, right? And so this is the first step of how people usually interact. And now let's say, oh, this study is very interesting to me. I can either save or like this article and then come back to it later for doing analysis, which I'll go in a while. But what you can also do is go back to the original source from which this data was got from. So you can go back and say, hey, okay, so it was posted in December 14th. I think there is a recruitment that is happening. I want more details of the study. You can go and get it there, right? But more importantly, now because we have gotten all of this information, we can also apply a lot of text mining tools on top of it. So similarly, we have different visualizations of how we want to visualize this data. We have the heat map view where what you're looking at here is the concepts, and what you're looking at here is the actual words in the article, and you can basically go over this, right? Now, of course, the easiest way or the most cumbersome way is I need to look at one by one article, right? But usually when researchers come into the space, they are basically looking for some way where I don't have to go through all articles, but I get a quick understanding of a group of articles, right? So for instance, one way that happens is, as I said, every single article in Tuxila is tagged. right? So you might come in and basically search for a tag. Let's say remdesivir, right? So every article
Sir,
which
sorry
has
for interrupting again, but the slides are getting stuck again and again. It's window size, you know.
okay.
It is stuck on the Taxila's homepage.
Okay. Okay, I'm going to try that again. So is it better now?
Yes.
Yeah, please let me know if it's stuck because I'm unable to kind of see that. So yeah. So as I was saying, so perhaps you also missed the part where I was opening a particular article and I want to read it. Of course, this is too long an article for me to read, but I want to quickly understand what are the different concepts. And Taxila has automatically applied its NLP algorithm and said that these are the most important concepts that this particular article is talking about, right? For instance, it's saying that it's talking about these pharmacological compounds. When you go in, it's going to tell you, okay, it's talking about dexamethasone, it's talking about remdesivir, it's talking about another drug, right? So the idea here is you don't have to read the article fully, but Taxila automatically summarizes that information for you, right? So you can go back, okay, which organic chemicals it's talking about, which amino acid peptides it's talking about, and so on and so forth, right? And similarly, as I said, one of the other ways Taxila is useful is, let's say you're interested in understanding remdesivir, right? So you can basically ask a question that I am interested in all articles that are tagged with remdesivir. And how many articles? So Taxila has already told you there are 28 articles in the last 1 month that have been published on remdesivir. So you can again ask a question. Oh, I'm not only interested in the last 1 month. I want to understand in the last six months how many articles related to remdesivir, how many articles in the last 1 year related to remdesivir, and overall, right? So then you're able to now say that, hey, in the last 6 months there are 35 articles that were published based on a drug molecule that I am interested in, right? Now the next question you could ask is, hey, I want to understand the trend of these 35 molecules because I want to come up with new hypotheses. So for that I want to understand different things about these 35 articles because I don't have time to go through each of these articles, let's say. And as I said, Taxila today has over 60, 70,000, or I think even more. I did not notice it. Yeah, 82,000 articles already collected in its system, so it can apply analysis on top of it. So one of the questions that typically people ask is, quickly, these 35 articles, can you summarize for me as a word cloud? So when I click that, what it does is it has automatically taken all these 35 articles, looked at the different keywords within these 35 articles which are talking about remdesivir, and it is telling you what is the lay of the land. So this is telling us that remdesivir is talked about with hydroxychloroquine, it's talked about with dexamethasone, and so on and so forth. And then you could ask, hey, okay, it's interesting, I want to know all the articles which are talking about remdesivir and hydroxychloroquine. So now if you see, it's talking about remdesivir and hydroxychloroquine, and you will see that these are the concepts that are talking about, right? There's a lot of interest in China. There is another drug seems to be implicated with them. And then if you go back, you will see that these are the 7 articles. So you can now zero down on which are the articles or publications that you want to work on, right? Now, another question that people typically ask is, hey, there are these 35 articles. I want to look at it from the point of view of the overall trends. So now Taxila has loaded these 35 articles and is creating a trend analysis of, over time, what are the different keywords based on which this particular data for remdesivir is coming. You can select different keywords.
Sir, sorry to interrupt you again and again, but I guess the screen is frozen again, I guess.
I'm so sorry. I don't know what is. Okay, but that is fine because I'll be anyway opening up the Tuxila subscriptions after this, so users can really. So is it better now?
Yes, sir.
Yeah. So if you see, this is another analytics module where it has taken these 35 articles and it is doing a keyword trend analysis. So it's telling you that, for instance, remdesivir, the trend of remdesivir in the last 6 months, what are the articles that have been coming into Tuxila? And recently, there has been a surge of these articles. So you can click on any one of them and say, okay, which are the articles which are relevant to remdesivir, which have been published in the last 1 month. And you can again drill down and ask questions about which are the other terms which are talked about together. And I hope that you did see the word cloud module that I showed, which kind of quickly summarized this to 35 articles, right? So similarly, you could ask another question that I want to generate a report of all these 35 articles, which are the what is happening, which are the all the chemicals that are talked about with remdesivir in these 35 articles, or which are the biomolecules that are being talked about, what are the pharmacological substances that are being talked about, what are the phenomena that are talked about. And again, when you click on them, you can drill down and get more details about each of these particular concepts. And you can also understand the geographical location, which are the other frequently occurring chemicals with remdesivir, which are the biomolecules that are talked about or diseases that are talked about, what are the distribution. So it has a reporting module. And another interesting facet is what we call as a galaxy module. So let me remove the remdesivir, and what you're seeing here is so let me reduce it so that it becomes more interesting. It is a galaxy, as we call it, where you're looking at the last 1 month worth of content that has come into Tuxila, and you're using NLP algorithms to understand how close these different articles are. So here every dot represents one article, and whether an article or two dots are close together means they're talking about a similar concept, right? So you could now visualize it very differently using different algorithms of how you want to visualize these particular documents. And if you see, you will see that there are clusters that are getting formed, and you will see that these are all talking about, each of these clusters are talking about very different concepts area. And you could now talk about, okay, within these articles, which are the ones that are pertinent to India, right? When I did that India, you see the dots reduced, and these are all the articles that are talking about India focus on in the last 1 month in the context of COVID. Then this particular set of articles is talking about very different things than this particular set of articles, and so on and so forth, right? And then another thing that we do in Tuxila is also ask questions around, hey, I have remdesivir as a drug. Now, what are the geographical trends of, let me say remdesivir. Let me go in on remdesivir ever, and I'm asking, where are all the clinical trials happening with respect to remdesivir? Now it's telling us that, look, in the space of clinical trials and for remdesivir, there are uh, trials happening in China, France, Germany. And when you click on any bubble, it's going to tell you, okay, which are the trials that are happening in China. And then you can go into the details of this one, when did it start, and all that, right? All of this information is now present in Tuxila. So here again, it's augmented intelligence in the sense that it is helping somebody, a researcher who's trying to work in this field, to come up with new ways of analyzing his data towards coming up with a new hypothesis or trying to understand the field better. And similarly, we could basically remove all of this and search, let's say, analytics or AI, artificial intelligence. So what it is going to do is, again, Tuxila is going to search within its set of articles and it tries to understand all the articles that are talking about application of AI in the context of COVID. You can actually select which set of articles you are interested in. But more interestingly, what you can also do is you can now say, I want to look at how the AI application is 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 scattered across different concepts. So for instance, here the search term that we gave is AI, and it is concentrated in different aspects, like here, where it's talking about a specific aspect, and on the other side where it's talking about a different application of AI, or a different facet of the application of AI in the context of COVID, right? So this space is telling us the different application areas. So this is just to give you a brief or a quick walkthrough of what you can do using text mining and text intelligence. Today after this talk, I'll be giving the access to these articles where students can go in and do a quick assignment of trying to come up with new hypotheses with this particular tool. And I don't know if we have time, but uh, we still have time, is it?
Yes, sir, we have time, sir.
Okay, so the next one I'm going to basically move into the space of what I said about getting the hands dirty, essentially. Um, uh, One second. Where the idea here is, okay, we've heard about all these applications of AI and everything, of NLP, and how it can be used in the biospace. Now, can we come up with can we get our hands dirty, right? For today's kind of a discourse, what I did was basically prepare a small um, programming kind of an assignment, or just to get started. And so this is again something that I'll share after today. So what you have here is essentially a piece of code which is going to try to analyze text articles that have been published in the context of COVID. So you have a metadata.csv file, and this file is actually a part of the CORD corpus, which was released by Alan AI for the scientific community to try to understand or make inroads with text. So this is something that I have already taken and packaged as a part of our lecture today. There will be a README file, which basically has the steps of how we want to get started. So it's basically based on Python. So all the participants eventually should have Python 3 installed their machine. A few dependencies in terms of a couple of libraries in Python that needs to be installed. And there is a requirement.txt file. And this file essentially has all the libraries that has to be installed. And again, I have put in details of how one has to install these requirements. And once they install all these 1 to 6 steps, it should be fairly straightforward. Then you go to your command line and type Jupyter Lab, right? So once this happens, what that would do is essentially open a page like this automatically, right? Open this a page like this, and what you're seeing here is an interactive Jupyter notebook. And again, since I did not want to use this workshop to give a lot of very complex concepts, but just a primer so that anybody who's interested can get started, some kind of a starter material to get started. So I have set up this thing, and people have to load this IPYNB file, which actually is a notebook, is a Python notebook, where you can see code along with the output of what you want to see. So you don't have to actually, so you can see and write annotations within the IDE itself, right? So, and I want to basically drive the point of how easy it is today to basically start working on text, because as I said, there are different communities, and there's a lot of research that's happening around text where a lot of the algorithms are being given to the community as something that they can freely use, and then the only thing is how do you then build up on top of these solutions to make a solution that is customized for your own problem that you're trying to solve, right? So in this particular and I hope you're still able to see it. Yes, is what we are doing is we are importing a library called Spacey, and Spacey is basically an NLP library which has a lot of the things that anybody would do as a part of text pre-processing already built. So all you have to do is import this, bring your data, and use these algorithms on top of it, right? So I'm importing Spacey. I'm importing some computational libraries called pandas. And within Spacey, there are different modules for visualization that I'm importing. So once I do this, what I'm doing is I'm reading the CSV file, and the CSV file basically looks like this, right? It's a big file. You can open it in an Excel too. And it basically has all the publications relevant to COVID, right? Now, what is interesting is a field called abstract, right? Because this field within the CSV has data, the text data within it, right? So once you read the file, let's say the first thing that you want to do is, let's say, show me the data in the 12th row corresponding to, I mean, the 12th abstract here. I want to know what is the text data here, right?
Sir, I guess the slides got stuck again.
All right. This is a recurring theme.
Yes, sir.
Okay. Um,
It's stopping your video might help.
yes. Thank you. So probably I should do that. Okay. So what you're seeing now is that I basically took this data set and I loaded a specific abstract. So this is the abstract that was there in the particular CSV file that has been given, right? So once I have this abstract, I can ask different questions. Okay. This is a piece of text that came from a publication. It's only the abstract. The first question is, hey, I want to do the classic what we call as an NLP pre-processing routine, right? Where what you want to do is extract the sentences. So as I said, again, using a language model, and again, this model is basically commoditized. It's there for anybody to download, is, and these have been trained on large corpuses of text. And once you download this model, you're now able to extract sentences automatically. So as soon as you say, hey, docs.sends, it's going to give you all the sentences and you can display it, right? Now, the next step is, given this particular first sentence, which is phospholipid vesicles containing bovine heart mitochondrial C oxidase, now do what is called a part of speech tagging, which means the algorithm now is going to take this whole sentence and it is going to say that this is the word by word phospholipid containing bovine heart mitochondrial cyto, right? Each one of the word is here, and it is basically saying what is the base word, right? So if you see here, it's called lemmatization, right? So if you see here, containing has been changed to contain, right? And it's telling whether it's a noun, it's a verb, whether it's a stop word, because I mean a stop word is typically like words like the, he, is. They'll be used a lot in the corpus and adding no value in that sense. So those information is all done, and again, thanks to this library, this is just about writing one line of code, and all of this text we have been able to look at whether there is a punctuation, whether there is.
Sir, I guess the site is again frozen.
Okay. Is it okay now?
Yes, sir. Now it is okay.
Okay. So the next step is, okay, so now I have basically done what we call as a pause tagging, where I'm able to look at the different types for every tag or every token or word that I have in my text. Now I want to do a dependency parser. So here what you're seeing is I'm taking this particular sentence and saying how is the dependencies happening. So for instance, this is telling that phospholipid vesicle is of a type compound. What that means is probably these two are together. Okay. And it is telling you what is the relationship. And as I said initially, when I was saying there is the word level, trying to understanding the meaning of the sentence. And this is where dependency parsers come into play. They basically take this text and try to understand the structure of the text towards getting to a meaning, right? So this is one of the, again, pre-processing step that people do. The next is, as I said, when if you remember in the Tuxila demo that I showed, when you take a piece of text, you want to identify different concepts within the text. So now I'm able to run an algorithm where I take this text, which is exactly the text, the single text that I took, and I apply an algorithm which is able to immediately say that, look, there is a mention of a percentage here, there is a quantity information here, all of this information like a person. I mean, again, here it has got it wrongly. It's gramicidin is probably a drug, but it says that it's a person. But it has gone ahead and what we call as in NLP as named entity recognition. It has gone and gotten these key concepts automatically. Now you will see that this is not very good for a biologist because all of this very good information is here, biologically relevant ones. And what this has picked up is very generic. And that's because of the model on which it trained on. So the next step, what I'm going to do is take the same text, but use another model. And this is the model called size spacey. And actually this model was specifically used by training biomedical or scientific text. Okay. Now you will see that by just importing this new model and doing the same thing that I did before, it's able to extract a lot more entities. I don't know if you're able to see it, but now it is saying that, look, in this piece of text, there are so many, the gray boxes are all corresponding to important entities which have some biological significance.
So we were not able to see the previous what you were showing.
Okay. Is
Yes,
it
sir.
clear? Yeah.
Yes, sir.
So here, if you see, I mean, it's the same piece of text, but by applying a model that was specific for biomedicine or biomedical literature, it was able to identify a lot more entities compared to a generic model here, where you see only few concepts were picked up. And the only difference I did was I moved to a different model, which was trained specifically on biological text. And you will see that it has picked up every single biologically relevant term as being important. Now, the question is great. It has found that these are important, but that's not going to solve my problem because I want to know what each of them mean. And that is, again, what you're going to see here is another model which basically takes the same text, but now gives categories to it. So for instance, it's saying phospholipid vesicles is a cellular component. Gramicidin is a chemical. And I don't know. Of course, there is a good chance that it also makes mistakes. For instance, cytochrome C oxidase, it says is a gene or a gene product. But here, if you see bovine heart mitochondrial C oxidase, it has wrongly tagged, right? So this is also the challenge of NLP in that I mean, different models have to evolve and be retrained specific for your specific problem at hand, right? So this was a quick kind of a demo where I wanted to maybe take you through what it means to read text, do a first level of understanding, and extract some key concepts from text, just like any other tool out there would do. And I'll be sharing this complete package along with a README of how students can utilize this with the presentation that we will share later today. But I'd like to kind of pause here and go back to my slide deck, and hopefully you will be able to see the slide deck. Are you able to see the slide deck?
Yes, sir.
Yeah. So as I said, we covered the part where we are talking about COVID Taxila and also an NLP primer, so that people who are now hopefully interested or inspired to get their hands dirty and use text as a modality, or consider as a part of their postgraduate or doctoral studies to look at text, to kind of start getting their hands dirty, right? And I think that part essentially concludes the part around text. And I think the next couple of things are, I think what Samik and our leadership team, Kitano-san, always says is, hey, it's great that by 2050, let's say AI systems can make scientific discoveries that are worth the Nobel Prize in Physiology or Medicine, which is basically the grand vision based on which we are driving. And this is actually a photo of the actual ceremony, Nobel ceremony. But the more pertinent question, in a funny tone, is apparently the Nobel will actually says that the Nobel Prize is for a person. And let's say there are changes made that AI can also win the Nobel Prize. But the interesting question is then, how can the AI attend the Nobel Prize ceremony, where they have to have the gala dinner and do the dance? So, I mean, we'll just leave you with that kind of a question, a funny connotation to this whole thing. But basically, that kind of concludes our discussions today. And as I said before, we are always interested in bright students and doctoral students to join us, both in the computer sciences and the more applied domain. And now I'll stop and I'll be happy to take questions. And a few links on all the things that we have shared today is available in the last slide. Yeah.
So thank you for this great presentation. Hope in next pandemic we will ask computer to give us the vaccine and computer will reply with an evidence code or a
Eventually,
protein to
yes.
target.
Yeah, we are
So,
very early in that game. Yeah, yeah. But
so yeah, in next pandemic, I think after 100 years. So that was a great session, and participants are very much liking them. So they have few queries. I would like to place them before you. So the first query is about the Garuda platform, that since different pipelines are based on different programming language, so do we get generic error message while executing a recipe, or do we have to make a way through the error logs? It is a
yeah,
bit.
that's a good question. And as Samik was also saying, the error logs are so as it was mentioned, there is the gadget or the software that you have, which is put into Garuda to enable recipe or the automation. Right now, the onus of the gadget being of good quality is on the developer. However, if you Garuda also keeps a track of all the logs, so it knows where, if there is a problem, that there is a problem that occurred. Then the user has to go and say, okay, the recipe may be stopped at a particular level because of a problem. Go and look at the logs of that particular gadget or the Garudified tool and see where the problem was. So yeah, I mean, the answer to that question is yes, recipe will tell you that there is a problem, but the user has to then go and dig deeper where there was a problem, and he or she has to use both the logs as well as their own intuition about the software module that was Garudified.
Oh, thank you, sir. So the next query is, how can we tackle the information bias that may be present in primary data? I think in context of text data. I think.
That's a very very pertinent question, and there are a lot of uh, um, there is a lot of body of work today to kind of look at this bias, right? Because when you're looking at this big space of all possible hypotheses and what has been achieved today, you also not don't want to be biased by what has been achieved today to look at what can be explored, right? And again, in this space, I think, and I think the whole field is moving towards that, is trying to today a lot of machine learning is around having teaching data where you come with your own bias. You say, okay, this is, so even in the demo that I showed you, let's say AKT1 is a protein, right? Now, how
Yes.
did the machine know it's a protein? Because it was taught, there was some training data provided where it was taught that, look, AKT1 is a protein. So any variation of AKT1 that you see, you tag it as a protein corresponding to AKT1, right? Now the whole field is also moving towards unsupervised, where you're looking at the whole corpus of text, not be very biased around how you want to organize, and this is more towards galaxy representation that I showed you. Here, if you see, we do not have any bias about where the text is coming. We are getting all of the text and looking at it from an unsupervised way. I mean, put it on a layout where there is a galaxy, and I will go and explore. So the short answer is, I think we are still figuring out, and I think there is no short answer, but I think these are the kind of questions which will drive the next wave of research in this field. Yeah.
Thank you, sir. Sir, one general question is that Taxila platform is only available for COVID, or any other
Yes,
diseases
yes.
of plant platform or
So
fungi?
at this point, we have made it available for the public for COVID. Of course, we use it for our other projects for different domains, but the public availability of Taxila is only for COVID at this point. Of course, the whole purpose of us developing Taxila is the application beyond COVID, but it's not publicly available. We use it for our own kind of research and work. Yeah.
Okay. And sir, one query is, can Taxila access the article which are not freely available?
Yeah, that's another very good question. So the COVID Taxila that you're seeing is freely available data that is there, that is open source, open. And the question around paywalls and whether you need access to a system, the answer is Taxila as a technology is agnostic to that. So if your institution already has a subscription, then Taxila can work along with it to get full text articles. But the COVID version of Taxila that you have is only for publicly available information.
Okay. And so one question is, I think from my side, that out of the curiosity, is that all this automation thing making the speed of research very fast, but how much manual annotation or manual review is needed? Like how much human intervention is needed for this textual
Yeah.
annotation?
So the answer to that is a lot, because as I said, it's great in terms of the progress we've made. We've come to a point where I think the next 5 to 6 years is going to be transformational. I mean, we might get to a point where, I mean, especially also with, I mean, if you see when Samik was presenting, he talked about AlphaFold, right, where it solved a big problem in the structure space, protein structure space. So, but it's still not there. It's still driven quite a bit by training where there is manual help needed. But having said that, I think today practically every data store that you have, there is a text mining engine that is helping in the annotations, right? So it's not perfect, but it has made significant progress. So of course now is to kind of look at the problems, like the first study where we presented that, look, there's 20 years of work, but it is still not come to a point where it is usable, right? So now where is the gap? So when we start plugging these gaps, I think, but again, I don't think it will ever come to a point where humans will not be in the loop. It's just that it is going to make us as humans more efficient, right? I mean, I
Of
think
course.
I think that's the main thing. I mean, there is no automation which is to replace humans. It's to make us better versions of ourselves, where I think AI will take us, is my thinking. Yeah.
See you, sir. And so one last question I will take. Can you give the names of the algorithm which you have used, like machine learning algorithms, which you have used to analyze those text data?
So, I mean, again, there is a big laundry list of algorithms that we use. But I mean, you could, even in the presentation that I shared with you, for tagging, there is a whole field of named entity recognizers. When you're looking at the galaxy, there is a whole space around computing vectors of words. So there is a work around word to wear, glove, and more recently, many of you would have heard about GPT-3. I don't know. I mean, the AI moment has come. So I mean, all of that essentially is in the
Also,
space.
BERT models are there.
Words, GPT, all of these are essentially things that solve different parts of the problem. So if you ask me a laundry list, the list just keeps growing. So,
Yes.
and I think that's also the key, as I said, right? I mean, the key here is how do you solve your problem with the tools that you have, rather than saying what are all the tools that are there. And We are also happy. I mean, a lot of the work that I showed on the research side is work that is published. So all the details of the algorithms and tools are there. But I'm also happy to kind of work with. I mean, if someone can send an email, I can share them all the kind of algorithms we are using. Yeah.
Okay. Thank you, sir. Thank you for your time and this nice presentation. We will share all your materials with the participants. And, thank you,
Thank
sir.
you.
So, participants, he was Dr. Sucheendra Kumar and Dr. Samik Ghosh, who have presented you this whole lot of knowledge about NLP. And there are a couple of announcements to make here that there will be one feedback link after this workshop, not every after every session. And for the next session, you have to download the tools like Autodoc and MGL tools. The link will be shared to you in the description section of the next session's video. And let's meet there. Okay. Thanks.
Thank you,
Thank you, sir.
everyone. Yeah.